Hi, my name is Olivia Yager. I'm a junior at California University of Pennsylvania studying molecular biology. During this semester, I had the privilege to study microbiology. Throughout our lab periods, we used the Tiny Earth program. The Tiny Earth program allows students to search for new antibiotics in the soil. The following presentation is an illustration of the various lab techniques used throughout the antibiotic search. Although my results lacked antibiotic producers, the techniques and protocols furthered my education and can be used by others to potentially find antibiotic producers of their own.
Starting off with soil sampling. Soil harbors an abundance of microbial biodiversity. Local soils are rich and dynamic. They also remain uncharted in the lab. The goal of the soil collection is to test the uniqueness of the soil and how much we can discover from the microbial life of the local sample. First, pick a sample that should be a good representation of rich biodiversity. This includes location, soil type, and weather. Then, using aseptic technique to collect the sample. Wearing gloves, I used my hands as a shovel and collected the sample in a conical tube. The tube was then stored in a cool, dark place until the next lab period for testing. Next were serial dilutions. To quantify the biomass of the soil sample, microbial counts were done. In most samples, bacteria ranges from tens of thousands to millions in such small samples. By diluting the sample using serial dilutions, the number of bacteria in the sample becomes more realistic to count. Start with 5 test tubes filled with saline. Then add a small amount of the soil to the first tube and mix well. Then add that same small amount of the first dilution of the soil to the second tube. Mix well and then repeat for all tubes. We only plate the last 3 dilutions in order to get a realistic number of bacteria to count. Plating, by using the spread plate method, is done by adding a specific volume of the dilutions to each new agar plate using a pipette. After the specific volume is added to the plates for the last 3 dilution factors, sterilize a glass hockey stick using a Bunsen burner. After it has cooled, use it to evenly spread the diluted bacteria over the agar. Then incubate plates to allow for growth. After growth, the bacteria can be calculated using the colony forming unit or CFU calculation. By counting the growth on the spread plates, the number can be entered into the formula to determine the CFU rate. Next, the spread plates were used to create streak plates and then pick and patch plates. The streak plate method was done first. It's used to isolate single colonies from the spread plates. 12 different isolates from the spread plates were used to make streak plates. For each isolate, you can do 1 streak plate, or you can do 2 isolates per streak plate, as long as you split it in half. After the streak plates incubated and formed single colonies, you can use the pick and patch method. The pick and patch method is used for preservation of each bacteria. This is done in case any secondary screening needs to be done. This method uses sterile toothpicks or loops to pick colonies from the streak plates and patch them onto the pick and patch plate. Next is recording colony morphology. Bacteria displays differences in physical appearance. The main descriptors are color, surface texture and elevation, shape, and margin. This is important to identify differences in isolates for further tests. Gram stains test for gram status in bacteria. Whether a bacteria is gram-positive or gram-negative can be important for determination of bacteria type. It can also help determine if a bacterial infection is present, what kind of bacteria is causing an infection, and can help determine treatment. The gram stain results purple for gram-positive bacteria and pink for gram-negative bacteria. The colored dye the bacteria absorbs is due to the membrane of the bacterial cells, called peptidoglycan. Using bacteria from the streak plates, place a small amount of bacteria on a slide. Dilute the bacteria and allow it to dry. After the bacteria dries, lyse the cells using a Bunsen burner. Then, use the stains in order, rinsing in between to successfully stain the cells. The stains in order are crystal violet, Graham's iodine, alcohol, and saffron. After the staining process, look at the cells underneath a microscope. If the cells are purple, they are gram-positive, and if they are pink, they are gram-negative. Next is the spread patch antibiotic production test. This uses the spread plate method and the pick and patch plate method together in order to test bacteria against a pathogen. The spread plate method is used first. You can do this one of two ways. First, by using the method used before with the sterile hockey stick and pipette. Or you can use a sterile swab to smear the pathogen all the way around the plate. Then, use the pick and patch method with the sterile toothpicks or loops to test all 12 isolates for antibiotic production. While this technique is simple, the implications are extraordinary. After incubation, you can check for halo formations in all bacteria. For each isolate, if halo formation occurs, that means an antibiotic producer is present. If no halo formation, then no antibiotic producers. In my case, there were no antibiotic producers. The next tests were the selective and differential media tests. These are used to further understand the bacterial status of our isolates. We used MacConkey and EMB, which is eosin methylene blue. The MacConkey only grows gram-negative bacteria. The EMB only grows gram-positive bacteria, meaning it selects for those bacteria. MacConkey stays pink for lactose fermenting colonies and turns yellow for non-fermenting colonies. On EMB, colonies will turn metallic green for lactose fermenting bacteria. For non-lactose fermenting bacteria, they will just stay normal. This differentiates between lactose fermenting and non-lactose fermenting for both MacConkey and EMB. The final tests that were done on our isolates were biochemical tests. The 3 tests done were the oxidase, the catalase, and the gelatin test. These tests are also done to further understand the status of our bacterial isolates. The oxidase test is done to test for cytochrome oxidase. This is done by applying bacteria to a filter paper and adding 2 to 3 drops of a reagent to the bacteria. Then wait about 60 seconds for the appearance of dark pink or purple color. If it appears, that is positive, and if there is no color, it is negative. The catalase test is done to test for the catalase enzyme. Apply bacteria to the sterile microscope slide and add 2 to 3 drops of hydrogen peroxide. If bubbles appear, it is positive, and if no bubbles, it is negative. The last of the 3 tests done was the gelatin test. This tests for the enzyme gelatinase. Acquire a nutrient gelatin tube and an inoculating wire. Sterilize with a Bunsen burner and allow it to cool. Add the bacteria using the wire to the gel. Stick it once through to the bottom and back up. After incubation, if the gel is still gel, it is negative, but if it's liquid, it is positive.